Hey, welcome back. Today we are talking about cell adaptation, one of the most important foundations in pathology. So, what are we going to be covering today? The first thing we'll be talking about is cellular adaptation. We are going to be talking about the big picture as far as cell adaptation is concerned. Then we move to the definitions that you need to know for each of these cell adaptations. Then we talk about the causes, the reasons behind each of these cell adaptations, and some of the factors that trigger these cell adaptations. The other thing we'll be talking about is mechanism, the how, what are the ways in which each of these cell adaptations take place. And the last but not least is to nail down the examples of each of these cell adaptations, which will likely be high yield as far as your exam is concerned, and also so that you can be able to recognize each of these processes in your clinics. Cell adaptation. So what exactly is cell adaptation? For us to define cell adaptation, we need to know some important characteristics of cell adaptation. One of the most important characteristics of cell adaptation is that cell adaptation is reversible. The other thing for you to know is cell adaptation is the structural response to stress. The third thing you need to know is that after cell adaptation, new but altered steady state of the cell is achieved. And the last but not least is cell adaptations allows the cell to survive and continue functioning. So basically, to define cell adaptation, the cell adaptation is a reversible structural response to stress in which new but altered steady state is achieved, which allows the cell to survive and continue functioning. So that is basically the definition of cell adaptation. It is a reversible structural response to stress in which new but altered steady state is achieved, which allows the cell to survive and continue functioning. So that is the definition of cell adaptation. What are the types of cell adaptation? So cell adaptations are divided into 5 main types. What are the types? 5 main types. The 5 main types of cell adaptation are hyperplasia, hypertrophy, atrophy, metaplasia, and the last but not least is dysplasia. So these are the 5 main types of cell adaptation. So the first type of cell adaptation that we will be talking about is hyperplasia. So what is hyperplasia? Just divide the word into two, hyper and plasia. Hyper means increase in number and plasia means cell. So look at this diagram here. You have a normal cell, one normal cell. After the cell has undergone hyperplasia, the cell divided into 3. So from 1 to 3, the cell has increased in number. So increase in number of a cell is called hyperplasia. So what is hyperplasia? Hyperplasia is defined as increase in the number of cells. Increase in the number of cells. So we now talk about the mechanism of hyperplasia. How does hyperplasia takes place? So in a normal cell, the membrane of the cell is made up of receptors. The receptors that are there, we call them growth receptors. So those growth receptors, when they come in contact with a growth factor, it causes the cell to grow. It causes the cell to divide. So the presence of growth factors and growth receptors on the surface of the cell will cause the cell to divide from one cell into many different type of cells. So one cell divided into 3 cell into 5 cell allows the cell to increase in number. So that is the mechanism of hyperplasia. What is the mechanism? Increase in growth factors and growth receptors. So now let's talk about the types of hyperplasia. Each of these cell adaptations have 2 main types, physiological hyperplasia and pathological hyperplasia. What are examples of physiological hyperplasia and what are also the examples of pathological hyperplasia? An example of physiological hyperplasia is increase in the number of cells in the breast and uterus during pregnancy. It's an example of physiological hyperplasia that occurs in women. An example of pathological hyperplasia is endometrial hyperplasia and also benign prostatic hyperplasia. So, knowing the definition of hyperplasia, knowing the mechanisms of hyperplasia, and also knowing the types of hyperplasia with their examples is something that is very high yield. So the next type of cell adaptation that we'll be talking about is hypertrophy. So just from the definition, or just from the name, we can derive the definition of hypertrophy. So hyper means increase and trophy means size. So hypertrophy is the increase in size of a cell. Okay, just to understand the concept better, if you look at this cell, this is a normal cell. The cell, after undergoing hypertrophy, that same cell increases in size. The cell became bigger, the cell enlarged. So increase in size of a cell is what we call hypertrophy. So the definition of hypertrophy is the increase in size of a cell. So what is the mechanism of hypertrophy? So the mechanism of hypertrophy is increase in cell organelles. We all know that inside the cell, we have organelles that are present inside the cell. The ribosomes, the endoplasmic reticulums. Those organelles increase in size and also increase in number. This is the result of hypertrophy. So when those organelles that are found inside the cell enlarge or increase in number, they cause the cell to get bigger. They cause the cell to enlarge. Hence, the process of hypertrophy taking place. So the mechanism of hypertrophy is increase in cell organelles. So what are the types of hypertrophy? As we said, there are two main types of hypertrophy. What are they? Physiological hypertrophy and pathological hypertrophy. Physiological hypertrophy and pathological hypertrophy. What are the examples of each of these type of hypertrophy? We start with the physiological hypertrophy. Physiological hypertrophy, an example is exercise-induced muscle hypertrophy. During the period of exercise, the muscle that is tend to be used extensively happen to get bigger in size compared to the muscles that are not extensively used. So increase in size of those muscles during the periods of exercise is an example of physiological hypertrophy. And also the other example that is widely used among students is the pregnant uterus. So during the period of pregnancy in women, the uterus increase in size. But there is something I want you to take note of. The increase in the size of the uterus is an example of both physiological hypertrophy and also physiological hyperplasia. So in exams, if you want to make things simpler for you, know that increase in the size of the uterus and also increase in the size of the uterus is an example of both physiological hyperplasia and also physiological hypertrophy. So now we move to pathological hypertrophy. Pathological hypertrophy, an example is cardiac hypertrophy in hypertension. So during the period of hypertension, the ventricles of the heart enlarge or they increase in size. So that is what we call cardiac hypertrophy in hypertension. So knowing the definition of hypertrophy, the mechanism of hypertrophy, the types and an example of each of those types of hypertrophy is high yield for your exams. So just to double down on what I was saying, these are two cross sections of the urinary bladder. So this is the bladder with also a normal prostate. So this is the normal bladder and this is the normal prostate. So if you look at the bladder has increased in size, so meaning the bladder has undergone hypertrophy. If you look at the prostate, the cells that are found inside the prostate, after the process of hyperplasia, the type of hyperplasia that takes place in the prostate is called benign prostatic hyperplasia. So after benign prostatic hyperplasia, the cells that are found inside the prostate increase in number. So increase in the number of the cell is what we call hyperplasia. So this diagram is illustrating both the concept of hypertrophy and hyperplasia. So the next step of cell adaptation that we'll be talking about is atrophy. So atrophy, what is the meaning of atrophy? So if we want to know the definition of atrophy, I want you to look at the diagram here. So if you look at this diagram, this is a normal cell. So after the process of atrophy, the normal cell has decreased in size. So reduction in the size of an organ or a tissue is what we call atrophy. So what is the definition of atrophy? Atrophy, this is the reduction in the size of an organ or tissue due to the decrease in cell size and number. So what is the mechanism of atrophy? So the mechanism of atrophy, as I said before, inside a cell or inside an organ, there are cells that are present there. Then inside those cells, there are organelles that are present. So those organelles, during the process of atrophy, those organelles will shrink, hence reducing their size. So the shrinkage of those organelles that are found inside the cell causes the whole organ to decrease or reduce in size. So the mechanism for atrophy is shrinkage due to the reduction in cell organelles. So knowing the definition and also the mechanism of atrophy is high yield for your exams. So what are the types of atrophy? Just like all other types of cell adaptation, atrophy is divided into two main types, and knowing the types and also an example or some examples of each of those types of atrophy is very high yield. So the first type of atrophy is called physiological atrophy. What are some of the physiological processes that happens inside the human body where our body part or a tissue in our body decreases in size? So the first one we have is called the thymus. The thymus, when we were young, the thymus is bigger in size, but as we grow older, the thymus tend to decrease in size, which is physiological. So the decrease in the size of the thymus in adult life is an example of physiological atrophy. The second one we have is called the ovary after menopause. After the period of menopause in women, the ovaries tend to reduce in size, which is something that is physiological. So the reduction in the size of the ovaries after menopause is an example of physiological atrophy. The third one we have is called the brain with aging. The human brain, as we get older, the human brain decreases in size. It tends to become smaller. So decrease in the size of the brain with aging is an example of physiological atrophy. And the last but not least is after the process of giving birth in women, the uterus tend to decrease in size. So knowing at least 2 or 3 examples of physiological hypertrophy is physiological atrophy, rather, it's very important for your exams. Now let's talk about the pathological atrophy. What is pathological atrophy? An example of pathological atrophy is starvation atrophy. So during the period of starvation, some parts of our body tend to decrease in size. So that is an example of starvation atrophy. Other types of pathological atrophy are ischemic atrophy, neuropathic atrophy, disuse atrophy, endocrine atrophy, and pressure atrophy. So knowing that different types of atrophy with example 2 or 3 examples is very high yield for your exams. So definition of atrophy is the reduction in the size of an organ. The mechanism of atrophy is the shrinkage due to reduction in cell organelles, and knowing the types of atrophy and also the example of each and every type of atrophy, it's very important for exams. So the other type of cell adaptation that we'll be talking about is metaplasia. So what is actually metaplasia? We define metaplasia as the reprogramming or the replacement of one mature cell type into another mature cell type. So when one mature cell type is replaced by another mature cell type, the process that has taken place is called metaplasia. So the definition of metaplasia is the process when one mature cell or adult cell type is replaced by another mature or adult cell type. So that is the brief definition as far as metaplasia is concerned. So one might ask, what is the mechanism of metaplasia? So the mechanism of metaplasia is the reprogramming of the stem cells that were present. So if the epithelium of a cell is having the columnar epithelium, and in the presence of stress, that columnar epithelium cannot withstand or cannot maintain itself in the presence of that stress. So in order to adapt to that stress, that columnar epithelium is going to be converted to a squamous epithelium. So the process that has taken place there is the stem cells that were present earlier in that cell are being rearranged into a different type of cell. So the process that has taken place there is called metaplasia. So once again, what is metaplasia? Metaplasia is the process when one mature adult cell type is replaced by another mature adult cell type. What is the mechanism of metaplasia? The mechanism of metaplasia is the reprogramming of the stem cells that are present. So now let's talk about the classification. What are the types of metaplasia? There are two main types of metaplasia. We have the connective tissue metaplasia and the epithelial metaplasia. So epithelial metaplasia and connective tissue metaplasia are the two main types of metaplasia. Now let's classify epithelial metaplasia. Epithelial metaplasia is broadly divided into two main types. We have the squamous metaplasia and columnar metaplasia, whilst the connective tissue metaplasia is divided into three main types: the osseous metaplasia, the cartilaginous metaplasia, and the myeloid metaplasia. So let's start with the epithelial metaplasia. The first one we have is squamous metaplasia. What is squamous metaplasia? Squamous metaplasia is a type of metaplasia when the squamous epithelium of an organ is replaced to squamous epithelium. So when the columnar epithelium of an organ is replaced to squamous epithelium, that type of metaplasia is called squamous metaplasia. So take note, the final product determines the time of metaplasia. So if the squamous epithelium is formed, the time of metaplasia is called squamous metaplasia. But if the columnar epithelium is formed, the time of metaplasia that happens is called columnar metaplasia. So once again, squamous metaplasia is a type of metaplasia that occurs when the columnar epithelium of an organ is replaced with the squamous epithelium. So what are the examples of columnar epithelium? The first one we have is called the bronchus in chronic smokers. So in chronic bronchitis, the smoke will convert the columnar epithelium that is found inside the bronchus into the squamous epithelium. So an example of squamous metaplasia is the bronchus in chronic bronchitis due to the presence of smoke. The other example is the gallbladder. Inside the gallbladder, we have stones that are there. We call them the gallstones. So those gallstones could convert the columnar epithelium to squamous epithelium. And the other one we have is called the prostate in chronic prostatitis. In chronic prostatitis, the prostate is converted, or the epithelium that is found inside the prostate is converted from columnar epithelium to squamous epithelium. And the last but not least is the endocervix. The endocervix in uterine prolapse, the epithelium that is found inside the endocervix is converted from columnar epithelium to squamous epithelium. And I want you to take note of this endocervix because the endocervix undergoes squamous metaplasia while the ectocervix undergoes columnar metaplasia. We are going to talk about it. So knowing the definition of squamous metaplasia and examples of squamous metaplasia is a high yield fact. So let's move to the other one. We have columnar metaplasia. What is columnar metaplasia? This is a type of metaplasia that occurs when the squamous epithelium is converted to columnar epithelium. So squamous to columnar, the type of metaplasia is called columnar metaplasia. What are the examples of columnar metaplasia? The Barrett's esophagus is an example, popular example of columnar metaplasia. The other one is the cervical erosion due to estrogen exposure converts the squamous epithelium to columnar epithelium, and also the stress due to chronic irritation converts the squamous epithelium in the ectocervix to columnar epithelium. So the endocervix undergo squamous metaplasia, but the ectocervix due to chronic irritations undergoes columnar metaplasia. So knowing these distinctions is very important for the MCQs because it is normally misunderstood or mixed by students. So the third type of metaplasia that we will be talking about is the connective tissue metaplasia, and the classifications are osseous metaplasia, cartilaginous metaplasia, and myeloid metaplasia. This osseous metaplasia, the most example is myositis ossificans, where a tumor occurs and the tumor is being replaced by a bone. Sometimes the cartilage also, some tumors are being replaced by the cartilage. And the myeloid metaplasia is when maybe during a particular process, the tumor or a particular pathology is being replaced by the bone marrow. So those type of metaplasias are called osseous metaplasia, cartilaginous metaplasia, and myeloid metaplasia. But the two most high yield types of metaplasia are the squamous metaplasia and columnar metaplasia. So knowing these facts are very, very important for your exams. So this is just a small diagram showing squamous metaplasia. This is a normal columnar metaplasia. If you look at the cells there, are columnar metaplasia. The columnar metaplasia is converted to squamous metaplasia due to the reprogramming of the stem cells that were present here. So what is the type of metaplasia? It's a squamous metaplasia. It is a type of metaplasia that is formed when the columnar epithelium is converted to squamous epithelium. So this diagram is just a brief illustration of the squamous metaplasia, and I hope it's helpful. So the last but not least of the cell adaptations that we'll be covering in this topic is dysplasia. So what is dysplasia? Dysplasia, if you want to understand dysplasia, we have to look at this diagram here. This is a normal cell and this is a cell undergoing dysplasia. Dysplasia simply is the disordered development of a cell. The disordered development of a cell is simply what we call dysplasia. It can either be characterized by cellular or cytologic changes. So the definition of dysplasia is the disordered cellular development. The disordered cellular development of a cell is what we call dysplasia. Simple definition. So what are the changes? What are the features that we have to look at for us to know whether a cell is undergoing dysplasia? So one thing for you to know is increased number of layers. When a cell undergoes dysplasia, the cell will increase its number of layers. One important thing for you to know. The next thing for you to know is disorderly arrangement of cells. So obviously, any cell undergoing dysplasia will have a haphazard arrangement of the cells. The cells will be disorderly arranged. The third one is loss of basal polarity. Basal polarity is having the nucleus of a particular cell lying on the base of the cell, like we have in these structures here. The nucleus lying close to the base of the cell. But if we have a cell undergoing dysplasia, that is lost. The nucleus are no more lying on the base of the cell. They can be lying on top of the cell. So the basal polarity that was there is lost. The third one is cellular and nuclear pleomorphism. Cellular and nuclear pleomorphism is the change in the shape of this cell and also the nucleus in a cell undergoing dysplasia. So the cell, the shape and the size of the cell changes. So that is what we call cellular and nuclear pleomorphism. The third one is increased NC ratio. NC ratio is the nucleus to cytoplasmic ratio. So if you look at the cell, the nucleus to cytoplasmic ratio is higher than that of this normal cell. And the other one is called hyperchromatism. Hyperchromatism is the nucleus is becoming more colored. Like if you look at the color of the nucleus here and the color of the nucleus here, the nucleus in the cell undergoing dysplasia is more colored. So that's why we said nuclear hyperchromatism. And the last but not least is increased mitotic activity. So there is more mitosis occurring in a cell undergoing dysplasia compared to a cell not undergoing dysplasia. So knowing that is very, very important. So these are some of the cytotolic changes that takes place as far as dysplasia is concerned. But one last more important MCQ question that is very high yield is dysplasia in all layers of the epithelium is called carcinoma in situ. This question is mostly asked in most of the MCQ questions in most of the university exams and also most of the board exams. Dysplasia in all layers of the epithelium is called carcinoma in situ. So carcinoma in situ is dysplasia in all layers of the epithelium. So thank you very much for listening to this video. If you are a new subscriber, please endeavor to subscribe to our channel and see you in the next video.